Hi, I'm Flaviu Bulat. I'm a senior research associate at the University of Cambridge, and I design, make, test, and translate into clinical trials novel imaging agents for the purpose of better predicting the outcome of any cancer therapy.
I'm Ahmed Mohammed. I'm a cancer research radiopharmacy manager in the Department of Radiology at University of Cambridge. My main research is focusing on development of production of 18C2M according to good manufacturing practices guidelines. Also, I'm a visiting radiochemist in Wolfson Brain Imaging Centre at the University of Cambridge.
So we're currently looking forward to a first-in-human study where we're assessing a novel imaging agent, injectable, that helps to predict the outcome of HER2 and triple-negative breast cancer therapies. So the injectable is called C2Am, and it's a small protein which detects cell death. And to help this protein visualize it while inside the body, we have made it radioactive by attaching a small fluorine-18 label. And by monitoring cell death post-treatment, we hope to be able to predict the outcome after just a single cycle of neoadjuvant therapy. So currently, patients undergo 6 to 8 weeks of chemotherapy before treatment outcome can be assessed, and we want to cut that down time to roughly 1 week. And this protein has been well established in the scientific community. We have used it in Kevin Brindle's lab for over 20 years now in animal models using different imaging modalities. And its use was demonstrated in more than 10 scientific papers. And after extensive toxicology testing and safety checks, we're now ready to take it forward to where it's needed the most, and that's the patients. So now Ahmed and I are going to walk you through the behind-the-scenes process of radiolabeling C2Am. We manufacture the radioactive agent in the Wolfson Brain Imaging Centre, more specifically in the Radiopharmaceutical Production Unit.
During gowning to the lab, we should wear a full gowning to protect ourselves from any chemical reagent that we deal with, as well to keep the environment as much as clean to protect our final product from contamination and, of course, protecting the patient.
We follow the guidelines of the Environmental Agency and the Health and Safety Executive for handling unsealed radioactive materials. And the basic principles set out by the legislation includes principles such as ALARA, as low as reasonably achievable. And in addition to that, on a daily basis, we apply TDS, so time, distance, and shielding, to protect ourselves from the high-energy gamma radiation. So this machine can accurately dispense radiopharmaceuticals for patients' injection, and it's doing so in accordance with good manufacturing procedures regulations.
We transferring a lead pot to the dispensing chamber to receive this dose. This lead pot will minimize the radioactivity exposure to ourselves and to the people that will transfer these doses to the patient or the other labs for analysis. As a part of dispensing in Thalia chamber, we need to transfer the dispensing material to the grade A, and this done by cleaning and transferring this material to a pre-chamber and doing sterilization for all these materials before starting the dispensing. This sterilization process helps to keep our product clean and make the patient more safe. The day after production, we need to do some cleaning procedures to the thinsis module. And before starting with this cleaning steps, we need to check the radioactivity exposure from the previous run. Each radiochemistry lab should have a Geiger counter that measuring the background activity, and this counter will give us a feedback if there is any increase in the background activity to take the safety actions in that cases. ENZ modular lab is a radio chemistry synthesis machine that we use for production of F18C2Am. This machine contains different parts. All these parts like valves and different sensors, pressure sensor or vacuum sensors or radioactivity sensor are connected. And there is a lot of lines connecting these valves that help in transfer of the chemical reagent that we used during the production. And all of these parts of the module controlled mainly with a laptop, as we did the programming for the chemistry sequence to ensure the repeatability of the production procedures.
So Ahmed is changing the QMA, which is a quaternary ammonium salt cartridge, in order to purify the final radiopharmaceutical. So the ENZ modular lab is specifically made so that it can custom for multiple radio tracers. However, this machine is now solely used by us in order to synthesize this specific radiopharmaceutical.
So we have a glass door to close the hot cell, and this will help to achieving the grade C area for the production. Then we close the hot cell, the main door, which is a lid door that will contain the all radioactivity inside and minimize exposure. And of course, we totally control the process using a laptop.
So the radioactive agent is being labeled in two steps. One step is making the prosthetic handle, and the second step is conjugating that prosthetic handle onto the protein within this automated system. And the process takes roughly 2 hours to complete. And once that's done, it's being transferred to the dispensing unit, which then delivers it to the specific vials.
So once we measured the radioactivity of the product, we print the label that contains all the data, time, and how much radioactivity in the dose. Then this label put on the lid pot. These data will be important for the patient when radiographer preparing the dose for injection.
So in the canister, we have one vial that's being delivered to the QC room, where it undergoes specific procedures to ensure the quality of the material. So QC stands for quality check, and it entails different procedures to ensure the stability and accuracy and specifications of the material.
So once we receive the QC sample from the production, we deal with this sample within a lead castle to minimize the radioactivity exposure. We firstly measure the radioactivity content of this sample, then dispensing the different QC vials that will be injected in different machines to complete the QC testing.
So this is an HPLC system. HPLC stands for High Performance Liquid Chromatography, and this is one of the 5 QC methods that we employ to ensure the stability and purity of the material. Others include gas chromatography, nuclide purity, gamma spectrometry, sterility, and endotoxin testing. The radioactivity levels injected into a patient are very safe, and they have been used for over 40 years.
So after finishing the all QC testing and have approval to release the product, the patient dose will move from the production facility to other imaging facilities like PET-CT or PET-MRI to start the procedures with the patient to be ready for injection and imaging after that.
So the C2Am project is considered a very successful preclinical project, and therefore is a potentially very translatable project. And in 2025, this will undergo first-in-human clinical trials.